Okay,
So
so right, I'm sharing because that's my screen. Anyone else?
Just a quick reminder, if anyone has any questions for our presenter today, please use the Slack channel we have within Slack for deep learning. We have a channel titled Local Talks, and feel free to drop any questions in there, and we will go ahead and answer those in proper time during the presentation. So if anyone hasn't accessed the Slack yet, does anyone need to get in? All right, well,
Okay, so great. Okay, so finally, it's my great pleasure to introduce Dr. Mulugeta work. For many reasons, not just because of the intrinsic interest of what he's going to talk about today and its relevance for our group, but Mulugeta is also a close friend. We were in the lab of Michael Bolver, where Mulugeta still works. As primate electrophysiologist at Columbia University for I think 6 years. And almost seven. Right. So we've been through a lot together. A lot of shared experiences there. I've come to know Mulugeta as not just a top shelf experimentalist, but also as an inventor. He invented a technique. Now, what's the patent status of that?
we have it now.
Right.
I'll mention it later.
So died in the patent for better fMRI registration of brain landmarks and MRI scans, right? And he came up with this, and people in the lab started using it. Columbia Business Office got wind, or brought it to them, and they're like, okay, we're going to go with this. And now it's actually a patent, which is not a trivial thing, right? Usually these days, patents happen, in a big company, and you have like 10 people, and you use some software tool, and then the lawyers get on it and push it through. But this was in this case, right? And I want a patent like that, right? And I have ideas on there, but it's just this big thing that's kind of an outgrowth of whatever dumb software project your company works on, right? But this is like this is old school patent, right? Where somebody has an inspiration, an idea, and makes it happen. So, in addition, numerous publications in the field of primate electrophysiology, and it's terrific to know that now Mulugeta is coming over to where the cool kids are, over to AI and ML, and that's what he's going to talk to us today. So take it away, Mike.
Thank you. Yeah, thank you, Matt, and everybody in the meetup, and RHT, Mike from LabCorp, everyone at RHT, but especially Morgan. Otherwise, I wouldn't be here. It's not easy to travel, long distance these days. Takes 2 hours of my time when I'm supposed to work. So today I'll tell you about what I've been doing in the lab where Matt and I've been together for many years. So many stories we're not going to tell you, maybe later. But we do actual neuroscience, and I think the reason why you got many people today is because people are really interested in what the brain does and how we do it, and why I got into ML and AI. And it's fun to share your data. So I am a research scientist at Columbia. I've been there for a long time. So the goal for me today is really to tell you about what I've been studying. It's called environmental memory, which is how we remember our environment, basically, how and why we study it, and why am I interested in AI, and how can it help us and other people, what my main results are. So by the end of my talk, you should be able to explain what environmental memory is. So there's going to be a quiz. I mean, in your house, when you tell the story, people will ask you. So you'll have to respond. And why are we studying it, and how, where are we going to go with it, and why we can use AI. So just to give you, an idea of why this is interesting, or, to just give you a frame of how to think about it. Most things in life and in the brain are all about networking. Not on purpose sometimes, but that's how things work out, how the brain makes sense of the world. And also there's some kind of trust involved in whatever network you are using. And there's some belief something will come out of it, which is exactly why I'm here today, because, network, trust, and belief. And really, anything you do in life ends up being, networking, or putting things together in your brain, or neurons working together, so we can answer different questions such as how do we get perception, sense, how do we move our bodies, language, how do we create it, how do we talk, how does it happen all together. And computer science has gotten to a level where now we say, how can we make computers become humans, basically, or the other way around, when you think of some people. So the way we look at this, or what I'm working on, is really related to how you see the world. Every day you go out, you see things, you want to make some sense of it. So ideally, you have some computations in your brain where different networks, different brain regions work together and come up with some kind of, solve some kind of solution about the problem you're facing. Let's say you want to move your eye, so we call that a motor output. Basically, you are just creating this, goal of doing something in your brain. In fact, there are some people who actually out loud say the brain is really all about movement. You move your hand, your eyes, everything that's happening in your brain. You move molecules or hormones. Anything you do has to do with movement. Uh, that's one way to look at it. But in general, the brain does something. So let's imagining this building. I don't know if you know this, some of you might. This is the Duomo in Italy. It's one of the best or the most beautiful things I've ever seen. So this is a piazza, there's a big region outside, it's a square. So if you lived there all your life and somebody took you to a lab and say, imagine standing in front of this square, can you describe it for me from memory? And this was actually done. So problem was, some people describe only half of it, and then they ask them, imagine turning around and now you're facing, your back is facing the church, can you describe it now? Now they will describe the other half, meaning they totally neglect half of their visual world. So this is usually when people have right hemispheric lesions. So today it's going to be a neuroscience talk, honestly, because the rest of it is not, problem for you, machine learning. But I will give you a long background on what we do and how the brain relates to machine learning, or maybe that's why you're interested. But in the end, there were a few papers who described, these basic problems, basically dealing with about 3 brain regions that are involved in problems like this: the frontal cortex, what's called parietal cortex in the top back of your head, and way deep in your brain called parahippocampal region. So if you have big damages around those regions or any big problem, there's a potential for this kind of very specific memory deficit. So who would have these problems? Which diseases cause this? And what don't we know about these problems is really what guides our research in our lab. So I sat down and made a big table like this, thinking about what problems are we facing with this kind of problems. So all the red ones are things we don't know and what we want to know. Mostly they have to do with schizophrenia, Parkinson's, anything that can cause you to have memory deficits or thinking things out loud in your head. So there's a big area of research in all of these different brain regions. Which side of the visual world is affected? How does this brain region work? Things like that. So just to come back to memory, this is a really famous but really bad definition of memory or classification of memory, because there's a lot of problems associated with it. But in general, there's short-term memory and long-term memory. Long-term memory is, where were you when, September 11 happened? And then you can actually describe what happened, where you were. And the other part is, things you do from memory, but you cannot really describe, like riding a bike, things like that, or things that you need to do your job right now, which is called short-term memory. Our memory is somewhere in between the two, which is it's a memory for things you don't really need now, but eventually they might become important. So our lab has found these neurons that seem to remember things that don't matter to you right now. And why would you do that? So any research you do in the brain is you basically train people to do some kind of task, and then you get to their brain and record their activity and see what's working and what's not working. Obviously, this requires a lot of hard work or many years of work because it's really complicated. Meaning you have to solve a lot of other problems, such as how do you target a given brain region precisely just from an MRI you take from the outside. So that's what Matt mentioned earlier, one of the problems, and I share this because it's important. So how do you visualize things that are not visible in the MRI? Basically, if I scan this, it will be black. You don't see anything. So I wanted something that I can paint over the head or this thing and then do MRI and know exactly where things are because I can now measure from those landmarks. And we wanted some flexible chemical. There was none. So basically we created it in the lab. So as a neuroscientist, you have to do lots of things, become a chemist also. So this is just one of the experiments earlier. So different kinds of MRI scans have different methods or reasons. So you do all kinds of experiments. In the end, we came up with this chemical. So I mention it also because it was extremely useful, and also we're actually looking for companies to buy it, and maybe LabCorp. Yeah. But Columbia is actually actively looking for, So in the end, this is really what neuroscience is. So the chemical you paint over anything, and you want to put your wire, that's an electrode that we insert into the brain. So we put this recording thing on top of the head. We acrylic it, basically, and then you go through each hole and find exactly where you want to go. But to be able to triangulate that, you have to know where everything is in space. So this is one of my electrodes recording from LIP, which Matt spent, many years recording from. So when you know exactly where you are, then you can look at the activity. And it's even more important when you go so deep in the brain. That's the parahippocampus. That's one of the areas I record from. It's extremely deep and dangerous because there are so many blood vessels and you have to be very careful. So to give you an idea of what neuroscience is, let's see. Or how do you do neuroscience? What is the daily activity? This is a screen. Somebody is moving their eyes, the dot moving. So they're supposed to move to the different corners, colors. At the meantime, the sound you hear, maybe you can't hear it because I have this on here still. Can you check the Slack if people have issues?
I don't have any Slack messages.
Okay.
But, I mean, I don't want to mess with audio. It's a working system now.
Okay.
If they miss, I will imitate.
Check.
Later for anybody who's missing it now. But nobody's.
Okay.
Sound is great.
Oh, okay.
Somebody's typing.
Okay. Good. So what you're looking at is the eye moving. We have wires or camera looking at the eyes, and it's moving. At the same time, we're looking inside the brain, and that's the eye being tracked. At the same time, that sound and the continuous activity you hear, I'll explain that in a little bit because this is really important. As I said, machine learning is not the most important here. But this is the brain activity, and you decide to give them different colors to call them different neurons. I'll also talk more about that. And as you can see, it's a very complicated process because everyone looks similar. So that's one reason why neuroscience can get complicated. And so what you do is, so we do, to record the activity, you first have to know how the neurons respond, like what's so unique about each neuron. So you do that by recording what's called receptive fields. Receptive field, by definition, is an area in what you're looking at that will activate one single ganglion cell in your retina. Basically, one cell has an activity. You don't respond to everything in the world, so each neuron will, respond to something. I will explain that again. So let's say you're looking at this screen. I ask you to fixate on the red square and don't move your eye. In the meantime, I want to find out an activity, let's say in this brain region, will give me 3 spikes per second, which is how fast the neurons are firing. Another region will probably, if I make it so small, or I just look inside that region, I'll get more activity. And in that particular region, I get 20 spikes per second. So we call this the maximum receptive or response field or receptive field maximum region. So from now on, I know your neuron that responds, I'm recording, responds to that location. So once I know that, I can give you a task like this one. This is called a memory-guided saccade activity. The first panel here is when you fixate on that red square, you don't move your eye. I flash something on the next block, which is the white square, because I know exactly where you will respond. And then you still don't move your eye. You keep fixating until the white and the red go out, which is the last panel. Now you have to move your eye to the remembered location. That's why it's called memory-guided saccade. So now I checked how good your visual response is, and also I will know how good your memory is, or that neuron's memory is. So this is called a memory-guided saccade. This is a very typical visually guided saccade or visual experiment. Then you measure the neural activity. So you measure it, given some time, how does the activity go up and down, and how can I calculate that? So this is, let's say, the whole activity of the brain going up and down, near that electrode. It's, just like, sound wave. We actually convert into sound wave. That's what you hear, chirping. But then you say, okay, if it goes above this threshold, I will call it a spike. Or every time it goes above here, I will just decide to call it a spike. So every time that happens, you do it. But now you can see 2 different colors, and there's a reason for that. That's because when the first one fires, I look back a few milliseconds, microseconds. So this whole thing is about 1.6 milliseconds. And I call that a neuron or a waveform. That's a very specific shape for this neuron. Another one will look like that. So we have a specific time to look back from this threshold and forward. Now we have 2 different neurons. And then it keeps happening. So every time I see that shape, I call it different neuron. So now we have 2 neurons. In 1 second, the green one, fired 6 times. So we call it 6 hertz or 6 spikes per second. The other one 5 spikes per second. And now, we say, what is this neuron doing? So which one do you think has memory? Yeah. Exactly. That's our problem. So how do we figure this out? What do we do more to find out just from this, or what, like this firing and things like that? So the next step in neuroscience is really, once you isolated a neuron, how would you go further down the road and say what to do with it? It's a very complicated process. As you can see, the shapes are similar.
So, if there are many neurons firing fast enough within the period of each other, is there a chance you could miss some of the firings?
Yes, depending on how fast you're recording, the sampling frequency. So most of this we recorded at 20 kilohertz. And depending on the instrument, the question you're asking, you can actually miss. And some neurons sometimes just become active in the middle of the experiment. There's a lot of things. But, it's hard. It's a very complicated work. Yes?
I'm trying to understand how you're actually isolating these neurons. Is it because of the temporal proximity and the refractory period? You're inferring that it can't be the same neuron because of refractory?
So most of it is template matching. So one is the shape, one is the amplitude. The activity is mostly what separates them. So you do quite a lot of things. Some neurons will actually have like a long refractory period, or they don't fire as fast, so you might miss them and they might come back in. Then there's something else. Yeah.
Can you just go back to that slide? I think you may have made it a little unduly difficult. Okay, so those shapes are actually quite similar, brown and green. They're not.
Well,
not
yeah, I could have made them. Yeah.
Okay, so but to the untrained eye.
Yes.
Right? Not like yours.
No, but I have another slide. I'll make that clear.
Right. So that secondary peak is different between the two most of the time. But in fact, normally the normal situation would be the shapes would be very markedly different.
I see. So this is pattern classification,
There,
possibly informed by temporal proximity.
yeah, there's quite a lot of things you could do. Yeah, we'll discuss this later.
Thank you.
Okay. But as this guy said once, he came from philosophy and math to do neuroscience. And I don't know if you remember this, but he said, what surprised me about neuroscience is really have to be extremely precise. And, he didn't realize, compared to math, which is surprising to me. But
How is math not precise?
yeah, but you have to come up with really ingenious ways and also really, yeah, exactly. But there is a whole field of neuroscience for every small thing. So there is a whole field of identification, signal identification. So there's this thing that this guy wrote. I'm still sore about a few things about it, but this is really my results from his software. I'm sore because he didn't sell it basically, and he had an offer. But you can see here, the black one and the green one are much different. So you can manually do this. There are automated processes, so you actually do more even after recording them. Okay, and we check different things such as are they excitatory neurons, inhibitory neurons. So this paper came out of Duke. I see a person with the Duke shirt, busy listening to me. So this is, you can check the shapes, one of your questions. You can have different measurements. So I actually used this tool also to check all of my neurons for the next part, which is parahippocampus. Let's go back to receptive fields. So whenever I move this thing around, some locations don't respond. There's no activity. That's what I was saying earlier. This location will cause 3 spikes per second. I move it yet again 10 spikes. I move it over there. So now your neuron is firing. That's where I want to focus. And every time that happens, I have a tick mark. So this is one experiment, one trial. So it happens. This is when I showed this white. So that's time 0. And then the next trial, you will do something similar. But not exactly the same all the time. That's the temporal difference in like the space between the two is not going to be always the same. But eventually you average it. You come up with what's called peri-stimulus time histogram. Peri-stimulus means around time 0. Some kind of stimulus, you average your signal around it and then say, when is the biggest activity happening? So for each brain region, this time might be very different. Some respond within 100 milliseconds, some 50, and I'll show you another one. Like this is my recording from parahippocampus, which was surprising at the time because most of that region responds way after 100 milliseconds. So we found out later where we were exactly just because of the timing. So every trial you see differences, but on average you see some repeated activity.
Quick question, is that time for the instrument to record or the actual...
For the brain to respond. Yep.
It seems slow to me in computer terms.
Yeah, so it depends on where you are. I have another slide later. So, the area I record from, which is the LIP, will about 80, 70 millisecond. There are regions in the parahippocampus that go up to 150 milliseconds, but it's sequential. Like the retina will be about 30 milliseconds or 20 to 40, and then thalamus will be around 45, and it's sequential. I'll show you another figure. So how do we look at this environmental memory, or what am I talking about? So that's the average I showed you before. You can do other things like standard error of mean, which is how distributed is this signal over time. These are different. And this is the eye movements, the horizontal and the vertical eye movements. So this is actually when the eye moves. How long does, whenever I place something in the, let's say, where your eye is going to move to, how long would it take for the eye to respond? And you can do different kinds of analysis. This is what we call baseline. That's before I show you anything. I record your activity and compare it to the visual response, or if there's some memory within this region, how does this compare to this? And you just make a ratio and, come up with something like that, some kind of memory index. You can do a lot of things. This is the first thing you do. And you can check different, subjects, different neurons. The activity may not look exactly the same, but it's repeatable depending on the experiment.
So there's different people thinking while you're doing the experiment, there's going to be a lot more activity in the brain, right?
Yeah, so that depends on what kind of experiment you're doing. But for things like this, when you're checking visual response or auditory response, those are precise almost all the time. Nothing really changed.
You can actually localize, get these kind of results.
Yes, but there are, some other questions such as attention, the intensity of the visual display. Those things do affect these things. There's priming sometimes, like if I tell you to look here, if I flash something, your response will actually be bigger. In auditory science, for instance, there is, like if you're looking at me talking, my sound actually is louder in your brain. That can actually be measured. If you're looking, so there are effects, but most of the main results are consistent. Otherwise, we can't even do science.
And which organism is this being tested on?
This is in primates.
Right,
Yeah.
Because you're actually doing a surgery to implant.
No, we do this in humans too. So people do this in humans. Yeah. Same experiment. It's almost the same result. I've done a lot of work in rats.
You're scaring people.
Yeah.
That happens in one specific rare situation, as we know, right?
Yeah.
When you're treating focal epilepsy, you have to put, and there's a wall between the clinicians and the scientists in this situation. So the scientific goals.
Yeah, I can't even. Yeah.
Electrode placement at all, but to treat otherwise uncurable epilepsy, they'll sometimes have to take out a piece of the brain, and before focal epilepsy, and so before they do that, they need to find out what that piece of the brain is doing, so they don't take out a speech center, right? And they'll put electrodes in for that reason, and that is the one rare case where we actually get.
Well, that's changing actually. These days, a lot of people are actually volunteering. Yeah, yeah. No.
I leave, and it's like Frankenstein.
Yeah. So do they know where you live? Huh? Don't tell them where you live, but.
Ask the audience for a volunteer.
Yes. So no, honestly, this is important discussion, but maybe for another time. But more people, more humans getting surgery for anything are now becoming, volunteers in research. In fact, I've been to one surgery that was so amazing to me. The person would not stop talking while his brain is being, manipulated. And I've been to another one, just like he said recently. I actually knew the person where they took out most of her, temporal cortex. So it's not going to be as scary as you think. So don't worry. So let's go back. This talk is going to go for 2 hours then.
Okay. All right. So let's
So how do we look at this environmental memory, or what it is, and how do I approach this? This is in general an approach guidance to people who do this kind of research or any research. How would I do it? Just giving you the rundown of my workflow, basically. So regularly, does that waveform or that average activity I showed you called PSTH, I look at the maximum, median, mean, depending on where I check. That's the first thing to start from. That's classical statistical neuroscience. But then, those points are distributed in some kind of distribution. So you check whether they're Gaussian or not, normal or not. In my case, most of those turned out to be not, which you do, with simple case test. So you do non-parametric tests. Basically, instead of t-test, you will do, other tests such as signed rank or rank sum, and you move on from there while you're doing the experiments, which are, in this case, let's say, if you're fixating here on the white square, now the receptive field, which we discussed earlier, would be the white area. So if you're fixating on the white, if I put anything on the white circle, your visual neuron, or what that neuron, let's say, will respond. If you move your eye to the red square, that location is going to move together. That's how you can make sense of things while you're walking or moving, because the eyes adjust where they respond to. So that we call that pink circle future receptive field. So the way I do my experiment is in different blocks. We call them blocks, meaning one block you'll do a lot of trials where you keep one thing, then the next block you change one thing, then you compare all those blocks. So in this case, let's say you're just moving your eye from this white to the red. There's nothing else on the screen, so you should not respond to anything. So that's your baseline activity. That's when you're sitting idle, just moving your eye. The next block, as I said, because when your eye moves the red square, the receptive field or your response field will move with it. If I push something there or flash something, now you'll respond to it. So my experiment, imagine mixing the two, where sometime I'll show you something on the future receptive field. Just like, you're sitting here, I'm here for a second, I'm not there for a second. So in the next round, when I'm not here, would your brain respond to it, to me, or my presence, or my former presence there? Meaning, so the best way or the easiest way to describe this, and I've said this many times, it's important because it's confusing. If you know there's a building when you walk to work, suddenly that building disappears, there are neurons in your head that will keep firing for some time, which is this next mixed experiment over here. The two experiments being mixed, where there's something when you're walking in the building was there, next time you're walking, it's not there. So in this instance, there was something in the last trial, but now it's not there. So there are neurons in your brain that remember that. This is important. This is what we call environmental memory, meaning you know where things are in space. And in this case, you don't need to remember it, but you do. So the question is why? And where does it come from? And it seems this is related to this people's problem where they cannot remember things. So if we look at the activity, again doing this in blocks, for your baseline, let's say you're responding about 5 hertz per second or about 3 hertz per second, the activity will just go up and down. Now I show you something on the future receptive field, your activity is going to go up to, let's say, about 13 hertz. That's an actual visual response. And if I do that only half of the time, it will still be as strong because there's something there. Now the question is, when I stop showing it, or in the trials where there's nothing, which is the pink here, I'll show you better ones later, but I like to put the median picture also, not to paint the perfectly cleaned up data, but the activity is still there. So that's the memory activity. Okay, you can do this for one neuron. So the magenta trace is what is the memory activity is like. You can do this for a lot of neurons, 49 neurons in one subject and 23 in another. This is the visual activity, which is obvious because you're responding to something. This is the pink one, is the memory. And if you do this kind of analysis where the x-axis is the baseline or when you're just sitting idle, and the y is when you're remembering things, you'll see that the activity is more on the memory side. And these are polyfit lines I put there to show you it switched the activity into a more stronger activity on the y-axis. You can compare the visual and the memory response. They're almost equal, but the visual is stronger. Again, to say that it's almost as if there is something there, but it's not there. So why would you do that? We know it's memory. This is because the timing is different from, remember you asked me, like you asked me the timing, or was it you, of the different brain regions. You'll get one time for one region, another. So that time is precise. If it is memory, it will be, much later than that. That's what this figure is showing. And it takes a lot of time to disappear. That's why I said, if the building disappears, how long does it take you to forget? So in this case, about 50 trials, the activity is still going. It goes down a little bit, but it's not completely gone. And I normalized it to the first activity that I record after I stop showing it, but it's still going. This paper is published, I think I shared it. Now, let's get to the problems, which is low yield. Meaning, this experiment, the whole thing takes me about sometimes up to 8 hours, 6 hours. In good days, you're shaking your head. In good days, if you're perfectly inside the brain and you get exactly what you want, 4 or 5 hours. The first part, recording the baseline activity when there is nothing happening, doesn't take long. To know where the visual response is, that may take 2 hours, 1 hour. So I should be done. Once I know that, I should just do the experiment and move on. But it's complicated. So the problem is, every day I record, and it looks perfect when I check that, mean activity, and I do statistical analysis, and they don't hold up. And my mentor comes in every day when we record, says, "Oh, great! This is a nice neuron. We should record this." And then the next day I say, "That neuron, no, it was not useful." So 6.5 years, basically. So there are false recording positives because they don't stand out through the statistical analysis. So the idea was like, why don't we first screen? As soon as I see their baseline in visual activity, I should be able to tell, if this neuron will have memory or not. You answered the green earlier, showed you 2 neurons. From that activity, if I was so good at telling which one is what, I don't have to do all the experiment. Plus, the next people doing this whole experiment should be able to say, oh, he said this is not going to have memory. In fact, I present this data many years, and every time people ask me, how come you're the only one who finds them, or how come you guys are the only one doing this experiment? And what is so special about these neurons? I say, well, I wish I knew that because it takes me a lot of time, and my yield is so low. So, finally I said, okay, I will do more deeper analysis and like really machine learning, AI kind of analysis, and be able to come up with some rule which tells me, this will be successful or not. Just like, predicting people's futures, ideally. So that's why I said I can decode the memory response from baseline and visual responses that I record early. And if I can do successfully that, then I will be happy. So every day, as I said, I record, neurons. Each in this, ugly spreadsheet, each row is a neuron. The columns are different, parameters I measure, median, max. It ended up being about 200 columns. Because every time, I talk to my mentor, we say, he'll say, have you checked, the median activity, I said, I checked about 200 milliseconds median. Tomorrow I'll add 150 milliseconds. So I keep adding. And people ask me, what is so special or what's important about these parameters? What's useful? This is exactly a tool for machine learning. Because if I don't do that, this is, simple approach, which you do correlation, and you find something so unimpressive like that. So if I use all of them, you can see some hot regions where there's some kind of activity or some kind of correlation, but this doesn't tell you a lot. So this is what the part I want to avoid. So if I could include all of the variables, all of those columns in my spreadsheet and predict memory and come up with a number with their prediction power for each one, and let's say convert it into word cloud like this, what do I get? The font size is really how predictive that thing is, that parameter is in my memory, but this is still a lot. Even though there are few big things that can predict memory, but if you notice, each of them have the word memory, which means I have used my results from the memory experiment to predict my memory, which is really totally the wrong way to do this. But it's a circular problem. So the goal is to go from that, use some kind of machine learning algorithm, and not use the memory experiment or the actual knowledge I have about those neurons, meaning the actual later knowledge I have, but use the early knowledge and then predict something that I can do and come up with something better and clean like this. In this case, you see visual response strength or how strongly they're, earlier I said 20 hertz, these guys can go up to 150, however, whatever region you record from. So if I can come up with something like that and small and take out all the other guys, that's really the goal. If you go home now, that's really the talk. Honestly, that's really what we want, and that's actually the result I got. That is, now useful to me. But the most interesting part for machine learning people is really how do you get there. So I will do, little justice to how the data approach. How do you do this? In anything you can use this, obviously you do, but I just formalized it. You clean up your data. I said I have 200 columns of things, 200 millisecond median, 150 millisecond median. Sometimes those end up giving me the same result. So I have to remove duplicates from that. And then check different machine learning tools and see which one will be really good at predicting what I'm looking at. meaning use all of my variables, predict, and then now boil it down to just a few important variables, because we get so much information in the world, but eventually we know what's useful and important. Why not do that for the brain? Then you can do all the machine learning things you do, classify, cluster, and know which one is what, and then predict. And once you have this very specific groups or variables or parameters, now you see how they relate to each other. And I'm going to rush through most of the approach because we're running out of time. But I will stop in some important places. But it's easy for you. Just to get back to what machine learning and AI is, it's really a tool for predicting something or using some knowledge to make machines do what humans do. And recently, the most famous one is deep learning, which is why we have this meetup. So how can we use that? So for some of you, again, to give you a primer, what's machine learning really? Machine learning is coming up with some kind of idea on how something goes from one thing to another. Even though you can't really formalize it or verbalize it, how things work sometimes. That's really machine learning, and it's just a tool to find an input and find that function that converts it into the output you want. Because what you do with it is you learn that association, how you can go from one thing to another. So in the future, when you get a new set of data, you can say, this is how to do it. Now you have a model. That's machine learning. And people call it different things, predictive analytics, predictive modeling. In my case, I wanted to check which ones are good. So the red here is most of the classical or the big groups of machine learning. So from each one, I tried to use at least one tool to come up with good solution for what I'm doing. And I don't know enough to explain to you. You can explain it to me better. But I'll just not going to go, I will not go through all of them, but the best one, or I will mention one that was extremely useful to me, which is decision trees. Decision trees are, in easy terms, they are how, without knowing much about what you're looking at, the results. How can you get into that result? You can go in different directions to one place, and how would that map look like? You just make if-else decisions in different steps. Let's say if I give you a bunch of images and you come up with some rule, you say if the images are colored, they will go on that side. If they're colored and they have only two colors, they will go on this side. So in the end, if I want to know which one is a machine and which one is a human, which is a 0 and a 1, how can I get there? So if I can get to the zeros and ones much earlier, that means I have a really good tree. I know exactly what it takes to be there. And that's really decision trees. And in my case, the ones and zeros are memory or not memory. How can I classify my neurons to get there? And there are many approaches. Variable importance is the first one that people use. It uses some kind of index where every time you have a new tree, you calculate how wrong you are in predicting what you're predicting, and then you do the tree all over again, see how good eventually you can get to classifying your data really nicely. And all you're doing is you're reducing your error, or what's called Gini index. For each branch, you just calculate how good you are. The error in misclassifying. Okay, back to our data. I mentioned first thing to check is really what tools are useful. In machine learning, there are so many. The 6 I tested are one of the top ones: random forest, classification and regression trees, LDA, PCA, SVM, KNN. So how good are they in predicting my data? And random forest and CART happen to be almost up to 100% accurate when they know the actual result in predicting my data. But obviously, I don't want to give the result to the classifier, so it's not going to be that accurate. But you test first what you can use. And again, you test different things, such as, how do you classify your data? In my case, 80-20 classification, for instance, turned out to be the best. You don't want to really overtrain your, your classifier, you just play with a lot of things basically. And the goal is, these 200 things I mentioned earlier, you rank them. So the top ones are really good at predicting memory, the bottom ones are not. So that angle is really what tells you. But you can see here, again, don't worry about this, specific names, but you see MEM everywhere, which is memory. That's again coming from the memory experiment in the whole data. So this is not my end goal. Again, this is there to, do some sanity test on my analysis and approach. This is the result that I wanted. Again, now you can see baseline in this V's mean the ratio between the visual response compared to the baseline, how good it is in predicting. You see baseline and visual everywhere here, meaning now I can use the baseline activity and the visual activity to predict my memory, and they rank in some order. That's machine learning, and that's why it's important. And then you can do more on, let's say, clustering your data. In this case, I chose 3 clusters randomly. Over there, I chose 10. They, visually that one looks better, but is that really good? There is an actual method to decide how many clusters you want to use. In my case, it happens to be 5. That's the optimal cluster. So right now, I'm just going through different, things, telling you how hard I work, basically. Not saying, it's all the best. Huh? Well, he got me here, which means he has done his job. Yeah. So you can do, PCA analysis, for instance. Like the most PCA is basically how spread your variance is, in which direction does it goes, or like what would explain the difference between us right here, in which, if we draw a line here, which direction is it gonna go? And so, we say, okay, the first line is the longest line within that distribution. What describes that? And then you have PCA2, which describes the other, perpendicular angle, basically. So in this case, I actually included the memory just to check what can the memory benefit. But you can see the baseline activity actually explains a lot of the variance on the first PCA, which was extremely surprising and useful to me. And this is again ranked. There's things called like external factor maps, how the different things relate to each other. This is gibberish, and it's not important, but you can see the thickness of the line is, how strongly these factors are related to each other. You can map all your relationships in different ways. This is a little easier to see. This is, called effect map. Here you can see 3 different things one time when you want to see some trend. This line, diagonal line, is going down that way. It's the actual memory activity, and compared to the baseline firing rate. For instance, you're all sitting here. If I record your brains now, some of you will fire at 300 hertz, some of you will be at 50. Matt will be, right now at 0, because he likes doing things, yes? But how about, the memory activity and then the visual activity? How does it relate? So the 5 different panels here are different visual, strength or visual activities. The y-axis is memory. So all the messages, every time the baseline activity goes really high, the memory activity is going to go down no matter how the visual response looks like. But this is not entirely true, because I told you visual responses matter. But the trend stays the same, okay? That's effect map. This is just correlation between the different parameters, how they relate to each other. This is just a logistic regression or Bayesian, logistic regression to see how memory and visual relate to each other. These are actually all of my neurons. I basically jittered them artificially so you can have the dots separated. Otherwise, they'll bunch up in 4 different corners. But you can see to have good memory, you'll need good visual response. That's the top over there. So that line tells you which way your regression is going to go using these different factors. I mentioned decision trees. This is one bad example. So the methods you use or the actual codes you use really matter. In this case, you can't learn much about it. This is, just H cluster, average cluster. This one is a little better. Now it's taking some of my parameters, putting them, in specific locations on the tree. but this is really the best one. Now, as I said, this is shorter, cleaner. The green and the pink are memory or no memory, and how can you get there? So in the top one says, if the visual response is, below, it's a p-value below this, it goes on this side. So you can get to the same thing in different ways, and you can now know how many neurons you have on that. So maybe you aim to get neurons, that are like that or this one. So any data you have, you want to know how to get there. This thing, people find it in GRC, like in a meeting, people found it funny and interesting because I lifted it completely off of market basket analysis. So some of the tools we don't really use in neuroscience, but there's always a communication. So when you go to the store to buy salad and spinach, somebody's going to think you're going to buy salad dressing. That's really what's called market basket analysis, and they use it. Whenever you buy stuff, they have a record of your member ID, what you bought, and people actually look at, how do we put things together in, this aisle, in that aisle, to, influence your decision. So in my case, if I have, neurons that have good visual and good baseline response, would that translate into memory response? And I use this because, you want to learn the associations from, low-level data and come up with some rules. So in this case, the algorithm found about 37 rules. The top one, or the stronger color there, is what's called lift, meaning it's strongly represented in the data. You're not telling it to learn anything else, but say, there's this data. What can you tell me about how these things relate? So it's just blindly associating things and saying this percentage of the things are happening together. So that's just analysis. So getting back to what we started with, there is visual input into your brain. You make some computations, you decide what to do. And Mike, you asked, if that's too slow. The retina responds about 40, milliseconds, and before you move your eye, there's about, 170 milliseconds passed. So this is really different, brain regions doing, one thing. The signal just cascades through the different networks and layers, and that's how you make decisions or you do some things. In this case, you get a visual input, goes to the thalamus, the back of your head. Some of it will go to the, the bottom of the brain, and then parahippocampus, these red regions, which I mentioned have issues with, people identifying things. This is, as you can see, it's a network problem, layered deep network problem. That's how, people think about identifying images or moving some things in the world. That's really why deep learning is so interesting to brain scientists and other people. How do you recognize images if it takes you that long? So I, being a neuroscientist, I lifted off Google's codes, as most of you do, because I don't have the time to write everything, but obviously you play with everything. So they have this, code that was written for digit recognition, which is an image recognition problem. And this is just a single layer version of it. You have an image, you have different weights and biases from each pixel value, and you come up with some kind of, transfer function which decides if the activity goes above certain level, or, if it just, stays below, nothing is activated. So whenever that threshold is passed, which is called activation function, you now have some kind of decision or some kind of classification. That's really deep learning. In my case, those parameters that I pulled out to be important, I put them together into a matrix and fed it into the network, and then say, can you tell me if there's a memory or not? You can do this in much complicated network. In this case, I used a 5-layer network, which was structurally like this. You have, an input layer of just the images, and then you have convolutional 3 different layers. This is exactly what they used. I just, have another layer there, which is called a dropout layer, for instance, which is at every stage of your training, you just randomly remove some neurons, which is the thing to do if you don't want to overtrain your network, which is basically this. And think of this as how you do things in your brain, because not every network will activate all the time. Not every neuron will do the same exact thing all the time. So you have to train your network to kind of, be in and out of, something to come up with a very useful, way. The next thing is you don't get the same input into your brain also. So there is a whole tool set for recognizing street signs. When you drive, some signs might be bent, rotated, faded. So how do you recognize? The brain does it easy. So people actually do that for image recognition. So I use just a few of those, translate the image, rotate it, do some kind of, uh, corruption of the data. So to get to the results, I can get up to 70 to 80% accuracy in predicting what the brain is going to do when it comes to memory using the deep learning and the baseline and the visual responses for these neurons. That's the conclusion of the deep learning part. That's the accuracy, and that's the cost or the error. You can do this in different ways. We can talk about that later. This is the whole network that I used. This is the augmentation part where I change the images in different ways and still get same kind of accuracy. That's the part that will interest you more, in the deep learning setting. But for me, how does the brain really require? I mean, how does it really recognize things, even though they're not the same all the time? So this region that we are looking at is called LIP. Actually has actual responses to your environment that may not be important to you at the given time. Even though those things move with your eyes, you somehow form a spatial memory of things. You move around in the world. You can even walk back to different things as if you know everything in 3D, and the brain must do that in spatial coordinates, we call it. And we know this memory is really there in your brain, and it's not an artifact of what I recorded. It's because we can predict it, and it cannot really be decoded from the data. So we still have more work to do, such as why do we have this? How does it work? And the next thing is, where does it really come from? As I said, 3 brain regions are involved in this. So for me or the lab, most of it has to go to the deep brain region, and I've been recording there. So we have to characterize that brain region. And because, as I said, everything works in a network, we do trust deep learning, machine learning, or the brain work in some way. And now we're testing our belief, which is giving it small data or early data to predict what it will do in the future. So these things always have to be useful or be done to predict the future. So this really puts AI and many things in our minds all the time. And why should we care? Because we think we can simplify what the brain does or how humans do things, and we can learn from it to solve actual problems without really being too cocky, saying we can do things, or the brain, this is really exactly how it works. That's really a bad goal to have. The tools work. As I said, we should not really trivialize things or our capacity. And who said this? I was in a meeting once. I was a reporter, and there was a bunch of neuroscientists asking this question. Can we recognize all these things and plan all the network, all the circuitry, and can we really figure out how this thing works? And this guy was a famous computational neuroscientist, Ken Miller, said, if we're so smart, we'd have figured it by now. And he's right, because it's complicated. You just have to use the tools for your purpose and advance the science, but it's not always easy. And someone told me, one of my mentors, this guy, he said, not everything we do matters now, and we don't know how impactful it is. So you just have to keep doing what you're doing, and eventually it will be useful. Same with deep learning. And this guy, who is my current mentor, says, don't be bogged down with everything all the time. Just do your work, because many things happen in life. Yeah, that's exactly who he is. Thank you. All right, so we should check the Slack for questions, yes?
Yeah, yeah, and I've been actually when you thought Mulugeta that I was like,
Yeah.
being a Russian bot on Facebook, I was actually monitoring the Slack channel.
Okay.
But the thing is, we had 2 people ask questions on the Slack channel, but I think both of them are here, right? I mean, Mike and your Justice, right? So why don't you guys just kick it off with your questions, and then obviously everybody can ask a question.
Sure. So one of the things that intrigued me during your talk was I was thinking back to this episode of House where one of the Foreman gets a communicable disease, but what it does is it attacks the brain, and part of the thing is extreme pain, but the other part is losing his losing his sight. But he still thinks he has it and can manipulate. He can actually walk around in his environment because he knows that he has this, what did you call it, retino,
Yeah.
basically a spatial model of where his environment, and he's thinking that he can see when he really can't. And so the question is, how much is sensory and how much is our imagination or model of our world in the way the brain is working?
That is a very, very nice question. In fact, nobody really knows if we even have spatial memory properly, or like purely spatial neurons have not been found yet. We know, there are neurons that fire when you're in a certain corner of the room or when you're turning different ways. They're head direction cells. But to actually, say, everything is coded purely in spatial, that has not been done. So usually you use a combination of, visual and spatial. But there are things you do, let's say in the dark. You can get used to things right away. You get to a new hotel room without turning the light on, you walk around. So we do that, but the percentage of how much is spatial is hard to know. And there are people, as you know, friends who don't know anything if you don't guide them, like people who cannot get used to new environments. So it's not a very trivial thing. But if you want to, let's say, help people who are like that, who become blind, can you, let's say, we're talking about this with Mike earlier, can you stimulate in their brain and tell them where everything is in space? We haven't gotten that far, and that's why we're doing this research, really. Plus, TV really, misguides us. It's also inspiring sometimes because, some of the things that Star Wars said, are now true,
So my first question relates to, in my own amateur research about EEG classification, I've encountered visually evoked potentials, like the P300 responding to a flash, etc.
Yeah.
What you're observing here in the receptive field feels analogous to that, even your statistical classification, averaging it out. What's different here, I want to make sure I understand, what's different here is how you localized the probe and isolated the actual effect, and it looks like it's a much lower latency response than what I've seen.
Yeah.
It's because it's averaged over the scalp.
Yeah.
Lots of neurons responding, or something that you take
Yeah.
with an EEG versus a neuron.
And this is a single LFP probe?
No, spike, yeah. Yeah, so there are different layers of, different levels of really studying the brain. So fMRI, for instance, is like global activity with blood activity, basically, when you do different things. And then you get to like magnetic activity. Electrical activity starts if you're recording EEG from outside the brain. And as she said, you're recording a bunch of neurons, but their overall activity, basically. It's like putting wire in a river, but you don't know, which water molecule is really causing the activity. So it's kind of a little gross. Like P300 and all those things are really timed events that happen. There's like ARP for audio, for instance. Those are really interesting. But if you really want to record pure neuron activity and you want to say something really about the brain, you get inside. So that's like very specific to the neurons, very specific to the brain region. EEG, even though you put it in a very specific, general area, it's not going to be so precise. But it's the same principle. You just getting more purified activity, basically.
Yeah, so you're answering the differences between the neuroimaging modalities, which I'm actually familiar with. I'm trying to make sure I understand would you consider this phenomenon you're observing to be an evoked potential? Is it truly analogous, even though it's like
Yeah,
more
it is in a way.
specific and less gross? It's
Yeah,
still analogous.
it's analogous, but it's very specific and like faster timed to specific activity, to specific neuron. It's just a better measure of what you're measuring. In fact, there are some people who don't believe in EEG, LFP, and we know a few people who would, yeah. But yeah, knowledge is really, as I said earlier, it's accumulated. So the fMRI knowledge actually guides like where to put your electrode. So it's not entirely wrong to do these things.
Differentiate, how do you deal with inter-experiment between subject versus within subjects? Are these calculations being done calibrated from a specific animal, specific brain, or are you drawing any background modeling from multiple experiments?
So it depends on what you're looking at. If you're working, let's say, on rats, you can do the same thing multiple times. Usually you will need more numbers, like 7 to 10 rats for same experiment. You go to the same brain region, the activity will look like. If you go to higher, like primates, you will do 2 at least. In humans, 1 is sometimes enough, but if you get a chance to record, you do 2 or more. So what you do is you repeat the trials, the whole experiment many times, and then you repeat different days. You can do many things, but if you want to really publish a good paper, you have to justify that this is repeatable. Other people can replicate the data, and you can't do that when you have only one sample. So there's an actual calculation.
You're not extrapolating to a different person or a different animal.
You are in a way, meaning whatever I recorded from one. So Bob Wirtz has a saying. What does he say? He's like, 1 primate is an experiment, 2 is a rule, or something like that. I forgot the exact phrasing. So usually, if you repeat it twice in 2 different subjects, it's going to happen everywhere. So
especially for something that's sensory, like the visual cortex, it doesn't vary a lot between primates and the people. So you can make those parallels.
Yeah. If you are asking like attention or consciousness or something serious, it's harder. But like hardcore, responses like visual, audio, movement, sensory, as she said, those are almost always there.
Monkeys are very expensive.
I think a lot of Ray's concern is mitigated by how you're registering to the receptive field at the beginning too.
Yeah.
So even differences between subjects are going to be mitigated because he's like isolating it to where is the receptive field in the electron.
Yeah, so depending on the brain region, like the one we record from, LIP, is one of the hardest ones to do because even the next neuron within like a millimeter will have an entirely different activity. That's why it takes very long to map these things. Some of the experiments I didn't explain because people ask, okay, if you're remembering this movement and the visual response look like this, what happens if I move this way? Which means now I have to redo my experiment fixating on the top of the screen, which means now I have to find another neuron that does something like that. Other regions, they have bigger areas that respond. So all neurons around, let's say 2-3 millimeters will probably look the same. So, like the spirihippocampus recording, I keep going down and I get the same response, the same activity. So there are, so it depends on the question in the area.
So MIT recently announced, and there was also a university out west, I can't remember whether it was California or Arizona, but that they're very high predictive rate reading your mind and recognizing words that you're thinking about. Are you familiar with that? And is the auditory brain signaling that much easier to machine learn than the visual?
I have to look at the papers.
Okay.
What do you think?
Well, why? So Mike posted a little screenshot of the device, and it's something that picks up on basically
phonemes, yeah.
sub threshold vocalizations and muscle contractions in
Well,
your
I see,
jaw.
Matt, I see the same kind of, we had somebody who had a electroencephalography, well, maybe it wasn't EEG, but I don't know. They were using it, and they called it a brain monitor, and they came and demonstrated Charlotte IoT, and they had the same sensor input behind the ear. Now, this does come across to the front side of the face, so they may be correlating the subvocalization, but the article didn't imply that they were picking up on subvocalization.
Oh, I'd really expect electrodes up here.
Yeah, and the device that we saw in Charlotte did have electrodes on the top, and it looked like they were grounding off the back of the ear.
Yeah, exactly. Mastoid bones right here is usually where you ground. So anything else would be recording.
Yeah, the only really interesting work I remember is from Georgia Tech, Kennedy's lab, where they actually want to do language prosthesis. They do implant, Utah arrays on people, and they try to decode what you're thinking, which is in phonemes, not words,
Right.
so they can vocalize it for you. That research is, almost successful. Like for language locked patients, when they have motor problems, like they cannot move their mouth. If you have, aphasia in the back, which is Wernicke's aphasia, that's hard to do because everything is gibberish, even for the reader.
Well,
So
I said there was a lot of surface level cortical pickup where they were actually doing imaging, and they've got imaging devices that attached to neurons, right, simulating imaging for simulation and the reverse process.
yeah, I mean, the way I can imagine it is if you train on given number of words, or like they give you a library of things to study, and they ask you to remember things and just think of them without saying them. Once you have the training data, you can probably, yeah,
Actually.
yeah.
By the way, you also used the term memory in your talk, and I was wondering if that was correlated to, or you were really talking about a thought versus a memory, or are the two that easily connected?
Oh, no, this is just pure memory.
Okay.
Yeah, so we're not thinking about,
you're actually
yeah, the neurons for the neurons. Yeah, yeah. Yeah, go ahead.
Actually,
Okay. Your motivating example about environmental memory, the piazza in front of the cathedral, that would be a difficult example to actually test for because there's so many variables to control for translation, rotation, distant memory, what mood they were in, lighting, and you've controlled a whole lot of things in this experiment. You've got them locked on a specific focal point, you know which receptive field, and you've even constrained time. It's a day in the lab, and you don't necessarily know how long it takes for that memory to decay. And I'm curious, how much of this you believe translates to environmental memory in the sense of remembering the piazza in front of the cathedral?
Yeah, so that is the next stage for our grants. I have one small grant where we want to do similar things in humans. So we haven't designed the experiment or anything. We're going to do it with the eye tracker that used to be in your room. So we'll track people's eyes. We'll give them different experiments. We can do like surface recording EEG or something like that. But the task is going to be difficult. Either you have to show them, let's say, a picture of like a piazza, degrade the image somehow, or do something that can really evoke memory. But the first thing for us was really to find, are there neurons who do this? But translating into human research, like on the field, we're in EEG and things like that, it's going to require patients who have, specific difficulties like this, and another cohort of, normal people. So,
My intuition.
There's no reason to say that what you've shown necessarily, even the very same region of the brain, it might be in a completely different region of the brain where the truly long-term memory is stored, right? You're seeing something on a time scale of seconds at most. And so he's asking, Well, environmental memory that we are familiar with is on a time scale of
Yeah.
days and weeks. But
Plus, almost anything we do in science and neuroscience, as I mentioned briefly, it's hard to translate things from here to here. So that's why I say don't overthink things, or don't think everything is perfect. It's going to be hard, but they are useful. You can now move on to the next step.
yeah, I did actually one.
Oh.
Just on your neural network, I think partially because of where I'm sitting, I didn't remember what was the input to your neural network. Can you just?
Yeah, so the input is once I clean out the important variables, it's
Yeah.
just, whatever it was that I was using, baseline, median, the ranked ones, those are changed into a pixel value, basically. So
Is it the topmost layers of the decision tree? Like the decision tree has a different.
yeah, everything above the bottom layer, which is, anything that I use to classify my neurons.
Okay.
So
it's just a feature vector.
exactly, yeah. And, I did submit a deep learning paper once, and one of the comments was, like, how can you do this? These are not images. But my response is, yeah, they are images. The brain, as long as it gets something, you have to do it.
But what was the structure of the network? This wasn't a convolutional neural network.
Yeah, so
It was a convolutional neural network on a feature vector.
Yeah, so the last one.
Well, now that's the kind of innovative thinking that Dr.
No,
Mulugeta
it came out of necessity. Most of my things happen because we need to do it,
So how did you make the square for the combinational?
Yeah, so like I pick, let's say, the top 25 variables or features that I like or that are good. So you just, convert them into a 2D matrix. So you just reshape it. Yeah, you reshape it into 32 by 32. Then you just feed it. So this you do this with everything, really, because I don't see why not. As long as I can tell, visual response is important. So the next people have it easier for our lab, at least.
I actually see reasons why not.
Yeah.
Yeah, go ahead. Yeah, let's see. Okay.
Yeah, it's because the placement in 2 dimensions of your features are going to be somewhat arbitrary, particularly when you go from row to row, and the kernel that you convolve with is going in 2 dimensions, and so neighborings that you didn't intend will be convolved together.
Yeah, so I can play with that. Yeah, which is the next step for me if I really want to push this. Like I can change the relationship between the neighbors, sort them differently, many things like that.
It might be interesting to convolve on 1 dimension with some sort of ranking. But
yeah, I haven't done that, but this worked up to 75-80%.
Yeah.
But that was actually like overextension of my authority as a scientist. Yeah. More questions?
So the other thought, sorry, the other thought is that for the neural network, at least for the first one, you could put all of the features in there and let the neural network decide which ones are not important, and then they get dropped out.
Yeah, so that decision I do it before the deep learning.
Right.
Yeah, so the next step will be, Yeah.
I know that from those surgical trials in humans, the famous observation was that direct stimulation of the brain is perceived as if it came from wherever it was supposed to come, but it was actually perceived to come a third of a second later rather than ahead of the natural stimulation.
Oh.
And they inferred the brain is doing a lot of post-processing with a date timestamp. And so I'm wondering how that
So you're talking about my PhD. So my PhD is in neuroprosthesis. Actually, the next step, which is, for hand, artificial hands, to create artificial sensation. Like, how can we do that? So my experiments where I, stimulate the fingers, recording the brain by manually stimulating, and then recording the different brain regions. So which digit is which exactly where. So then I go in and put electrical current into the brain, trying to recreate that. That's exactly the problem with.
subjects, which came first, that was liver.
No, like you just look at the brain activity and say, does it look like when I was touching, this one. So right now the level of the research is people can actually tell which fingers is being touched, or I mean, these normal subjects, and also, amputees. You can stimulate and still create which direction to move, things like that, depending on the frequency or the location you stimulate. So it's not as bad, but it's not an easy thing to do. Especially if you're moving your arm, you don't look at it. For instance, when you look at things and you touch them, the signal gets better. So there's a whole lot of problems. But the signal doesn't take that long, no matter what you do. It's actually much faster than that. Yeah, he's one of the really old people in this. Yes. Yeah, I quoted him many times. Yeah. Yeah.
So on your decision tree, are you just reweighting it, or can you also reshape your decision tree? Can you move decision branches?
Yeah, so this is randomly activated, so it does it, automatically all over again many times before it gets to a conclusion.
And the other question I have is, it seems like reading the brain is a matter of discriminating superposition of a whole bunch of signaling, right?
Yeah.
So, do you have? I saw in your PCA analysis baseline, and I'm thinking you're going to take a lot of signaling out and basically you can focus in on what you're looking for, right, in terms of your memory. So do you look at this whole? And one of the questions I had is we have like in CUMC Fourier analysis, where you could take a particular frequency out and look at it and use that for determining something. Is there a Fourier analysis of neural signaling?
Yes. Oh, yeah, a lot. So like the EEG signal is mostly done on Fourier space, basically.
Okay.
And LFPs, local field potentials, because they're like, there are things you cannot do with frequency. Like one time I did Granger analysis for the two brain regions that I was recording from. You can't do that with frequency data because it gives you wrong results. But there are people who actually spend all their lives looking at frequency communications between different brain regions. In fact, some people think that's how we make decisions.
Well, you mentioned the good-bad discrimination was partially shape, and I'm thinking that there's a frequency basis for your shape, right? And perhaps some of that could be discriminated just at the Fourier, just at the frequency component analysis.
Yeah, so the frequency can be useful in some ways. So there are two things, at least for sensory perception. People say it's the timing of like how the spikes happen. Other people say no, it's the frequency. You just have beams where you just do frequency analysis. In fact, that's the theory that wins the most because people can now discriminate different frequencies, and the brain seems to make decisions based on frequencies. But that's a whole. You had questions for a long time.
Yeah, so let's let this be unless maybe these two questions be the last ones. We have the room for about 6 more minutes, and we want to kind of have a chance to rearrange things. So you guys are going to close it out here.
Yeah. So the question is, this response, this memory response, is happening much later, or comes out much later. What's the source of it?
We don't know.
So is it, what all of the sources are for the input to that area, and then, how might you...
Yeah, so the reason why we went to parahippocampus is exactly that. The LIP is not a memory region. It just is a region which makes you decide on what's important in the environment. But it must get this memory from another region. And the one that's connected to it and has memory function is parahippocampus. That's why I went there. But I wonder who you are, and if you want to say words. And also, I would like to get feedback on what you guys think about deep learning and brain.
Okay. No, but there was one more question.
Yeah.
Did you do some research with spatial brain, like brains with ADHD?
Not yet. Yeah, but these are the right people to, yeah.
All right, let's give a big round. You guys can talk. And
Okay. Thank you, everyone. I guess we didn't get enough time to answer everybody's question.
No.